Welcome to the deep dive. Today we're tackling a really neat biological puzzle. Picture this. An animal that looks, well, almost exactly like a snake, moves like one, but it isn't actually a snake.
Ah, you must mean something like the European glass lizard.
Exactly. It's legless, it slithers, but if you look closer, things are different. Its jaw isn't mobile like a snake's. It has fewer vertebrae, a different tail structure.
Right, it tells a completely different evolutionary story. It belongs to a totally separate lineage from true snakes.
So that raises the big question, doesn't it? How do biologists possibly sort through the millions of species on Earth and figure out their actual family histories, especially with tricky cases like that?
That's really the core of what we're diving into today. Think of this as your shortcut to understanding biological classification and tracing those evolutionary relationships. We're drawing straight from a key chapter in Campbell Biology in Focus.
And our mission really is to unpack some pretty dense concepts, phylogeny, classification, even things like molecular clocks, and make them clear, make them stick,
Absolutely. And the central idea holding it all together is phylogeny.
which is basically the evolutionary history of a species or a group of species.
Precisely. And understanding that history, well, it unlocks some really surprising connections and helps make sense of the sheer diversity of life out there.
Okay, so where do we even begin trying to categorize all this? Maybe with names.
That's a great starting point because common names can be a real mess. Think
Okay.
about jellyfish, crayfish, silverfish.
You hear fish and assume they're related, right? But they're totally different.
Exactly. A jellyfish is a cnidarian, crayfish a crustacean, silverfish an insect. Nothing fishy about them, biologically speaking. Common names cause confusion.
So scientists needed something better, more precise.
Which brings us to Carolus Linnaeus back in the 18th century and his binomial nomenclature, the two-part scientific name.
Like Panthera pardus for the leopard.
Perfect example. Panthera is the genus that groups closely related species like lions, tigers, leopards, and pardus is the specific epithet, unique to the leopard within that genus.
And there are rules, right?
Yeah.
Genus capitalized, all italicized.
You got it. Standardized, clear communication. And Linnaeus, maybe a bit optimistically, called us Homo sapiens wise man.
Wise man indeed. Okay, so he gave us names, but he also organized things, didn't he?
He did. He developed a hierarchical classification system. It's like nested boxes or maybe a biological postal address.
Taking you from specific to general.
Right. Species, genus, family, order, class, phylum, kingdom, and now we add domain at the top.
So for our leopard, Panthera pardus.
It's in the genus Panthera, the family Felidae, that's the cats. Then the order Carnivora, class Mammalia, phylum Chordata, kingdom Animalia, and domain Eukarya.
Wow, quite the address. And each level is called a tax.
A taxon. Any named unit at any level. Linnaeus basically built the first universal filing system for life.
Okay, that organizes things nicely, but it doesn't automatically show the evolutionary relationships, does it? Like who descended from whom?
No, not inherently. That's where the phylogenetic tree comes in. It's a visual hypothesis, branching like a family tree, showing those evolutionary connections.
So the points where the tree splits,
Those are important.
vary. Those branch points or dichotomies represent common ancestors where lineages diverged.
Like the point where the human line and the chimpanzee line split off.
Exactly. And humans and chimps would be called sister taxa on that tree because they share an immediate common ancestor that other groups on the tree don't.
And sometimes you see trees with one main trunk at the bottom.
That's a rooted tree. The base represents the most recent common ancestor of all the taxa shown in that specific tree.
Now here's something that really blew my mind. These trees show descent, not necessarily how similar things look, right?
That's a critical point. Take crocodiles. They look way more like lizards than birds, wouldn't you say?
Definitely. Scaly, four legs.
But phylogenetically, crocodiles are more closely related to birds. Birds just underwent a lot of evolutionary change. The tree tracks shared history, not just outward appearance. And it's not like chimps evolved from gorillas, or humans evolved from chimps walking along a line.
No, absolutely not. Taxa next to each other on a tree share a common ancestor further back in time. They diverged. It's about branching, not a ladder of progress.
Okay, this is fascinating stuff, theoretically.
Yeah.
But does building these trees actually help us, in the real world?
Oh, absolutely. Phylogenies have huge practical applications. Think about agriculture.
Like with corn.
Exactly. Understanding the phylogeny of wild grasses related to maize helped researchers find relatives with useful genes, maybe for drought resistance or pest control to improve our crops.
That makes sense. Any other examples?
There's a really striking one involving conservation, illegal whaling.
How did phylogeny help there?
Investigators found whale meat being sold, but they weren't sure which species it came from. Some are highly protected, others less so.
So they used DNA.
Right. They created gene trees from the meat samples. The position of the sample's DNA on the phylogenetic tree told them precisely which species it was, even identifying illegally harvested endangered whales.
Wow, so it's like biological forensics, providing hard evidence based on evolutionary relationships.
It really is. Shows how powerful understanding relatedness can be.
Okay, so to build these powerful trees, we need clues. We need to compare organisms. But how do we know which similarities actually matter? Because sometimes things look alike just by coincidence, right? Like our glass lizard and snake.
Precisely. This is the crucial distinction between homology and analogy.
Homology is similarity because of shared ancestry.
Yes. Think about the bones in your arm, a bat's wing, a whale's flipper, the underlying structure, one upper arm bone, two forearm bones, wrist bones, finger bones. It's all the same basic plan.
Inherited from a common ancestor way back when.
That's homology. But then you have analogy. That's similarity due to convergent evolution. Different ancestors, similar environments leading to similar solutions.
Like those burrowing mammals.
Right.
The Australian mole and the African golden mole.
Perfect example. They look incredibly similar on the outside. Strong limbs for digging, tiny eyes, protected noses. You'd swear they were close relatives.
What or not?
Not at all. Internally, genetically, they're worlds apart. Their last common ancestor lived maybe 140 million years ago and definitely wasn't mole-like. They independently evolved those burrowing features.
And the glass lizard's leglessness is like that too, convergent with snakes.
Exactly. An amazing example of analogy, driven by similar lifestyles, not close kinship with snakes. Distinguishing homology from analogies is fundamental to building accurate trees.
So how do you tell them apart? Is there a rule of thumb?
Generally, the more complex the structures you're comparing, the less likely they evolved independently. Think about human and chimpanzee skulls, the number of bones, their shapes, how they connect. It's incredibly intricate.
The odds of that happening twice by chance seem tiny.
Exactly. It strongly suggests homology. Same idea with the DNA. If two sequences share long stretches of nucleotides, especially in complex genes, that's strong evidence for homology.
Okay, but DNA changes over time, right? Insertions, deletions. How do you compare sequences from species that diverged millions of years ago? Must be tricky.
It definitely is. Sequences can get misaligned. That's where computers are indispensable. Special programs help align the sequences, sometimes adding gaps where deletions or insertions likely occurred to find the genuinely corresponding homologous regions.
And these molecular comparisons can reveal relationships even when appearances are deceiving.
Absolutely. Like the Hawaiian silver swords. They look incredibly different. Some are shrubs, some low-growing rosettes, but their DNA is remarkably similar, showing they all evolved relatively recently from a single ancestor.
So once we've got our homologous characters sorted out, how do we actually assemble the tree? What's the method?
The dominant method today is called cladistics. It focuses strictly on common ancestry to group organisms.
And it groups them into clades.
Right, a clade is the goal. It's a group that includes an ancestral species and all of its descendants. It's a natural, complete evolutionary unit. Think of the cat family, Felidae, that's a clade. All cats descended from one common cat ancestor.
And Phylogeny is itself part of a bigger clade, Carnivora.
Exactly. Clades are nested within larger clades. And biologists really aim to define these monophyletic groups. That's the technical term for a clade.
Are there other kinds of groups that maybe aren't so good?
Yes, and we try to avoid them because they don't accurately reflect evolutionary history. One is a paraphyletic group.
Para, meaning beside or near.
Kind of. It includes an ancestral species, but only some of its descendants. The classic example used to be reptiles, excluding birds.
But birds evolved from reptiles, right? So leaving them out makes a group incomplete.
Precisely. It's paraphyletic. Then there are polyphyletic groups.
Poly meaning many.
Right. This group's distantly related species but misses their most recent common ancestor, like maybe grouping seals and whales based only on their streamlined bodies.
Ah, based on an analogous trait, not shared ancestry.
Exactly. So cladistics focuses on finding those true monophyletic clades.
Okay, so to build a clade, what kind of characters are we looking for?
We need to distinguish between two types: shared ancestral characters and shared derived characters.
Ancestral versus derived. What's the difference?
A shared ancestral character originated way back in an ancestor of the whole group we're looking at, but it's also shared with other related groups, like the backbone in mammals.
All mammals have one, but so do fish, amphibians, reptiles. It's ancestral for vertebrates as a whole.
Exactly. But hair that's different for mammals. Hair is a shared derived character. It's an evolutionary novelty that's unique to the mammal clade, setting them apart from other vertebrates.
So derived characters define the clades.
That's the key idea. And importantly, losing a character can also be a shared derived character. Like snakes losing limbs, that's a derived trait defining their specific lineage within reptiles.
How do you figure out which is which, ancestral or derived?
We use an outgroup. That's a species or group closely related to the ones we're studying, the in-group, but known to have diverged earlier.
Like using a lancelet, which isn't a vertebrate, to study vertebrates.
Perfect. The lancelet lacks a backbone, hinged jaws, limbs, etc. By comparing the in-group vertebrates to the outgroup lancelet, we can infer that features present in the in-group but absent in the outgroup must have arisen within the in-group lineage. They are likely derived characters for that group.
So you map these derived characters onto the tree, showing where they first appeared. Backbone here, jaws here, limbs here.
Exactly. Step by step, building the branching pattern based on shared derived traits.
Now, looking at these trees, sometimes the lines, the branches, have different lengths. Does that mean something?
It often does. Branch lengths aren't always just arbitrary. They can be scaled to represent something.
Like,
Two main things. Sometimes length indicates the amount of genetic change. A longer branch means more mutations have accumulated in that lineage's DNA since divergence.
okay, and the other?
Chronological time. If calibrated with fossils, the branch lengths can represent actual time duration. In that case, if you trace from a common ancestor to all living descendants, the total path length should be the same for everyone.
Because we've all been evolving for the same amount of time since that ancestor lived. Makes sense.
Right. Different ways to convey information in the tree structure.
Hold on. If you're comparing, say, dozens or hundreds of species, there must be, well, billions of possible ways to draw that tree, right? How on earth do scientists choose the best one?
That is a huge computational challenge. And the guiding principle they often use is maximum parsimony.
Parsimony, like being frugal.
Or,
Simple.
exactly. Think Occam's razor. The simplest explanation is often the best. In phylogenetics, the most parsimonious tree is the one that requires the fewest evolutionary events to explain the data.
So the fewest appearances of new traits, or fewest DNA-based changes.
Precisely. Computers analyze the data and compare the vast number of possible trees, identifying the ones that required a minimum number of evolutionary changes, like mutations or morphological shifts, to get from the ancestor to the observed species.
So it's about finding the most efficient evolutionary path.
In a way, yes. It assumes that complex pathways with lots of reversals or independent origins are less likely than simpler ones.
But it's still a hypothesis, right? Not absolute certainty.
Absolutely critical point. Phylogenetic trees are always hypotheses. They represent the best fit to the current data, but new data, more DNA sequences, new fossils can lead to revisions. They're dynamic.
And this hypothesis framework allows for some cool predictive science, like phylogenetic bracketing.
Yes, this is where it gets really exciting. Phylogenetic bracketing lets us make predictions about extinct organisms based on their living relatives.
How does that work?
You look at an organism's closest living relatives on the tree. If those relatives share a particular feature, say a behavior or a physiological trait, you can predict that their common ancestor and likely other descendants, like our extinct organism, also had that feature, unless there's evidence otherwise.
A dinosaur example is amazing here, isn't it?
It really is. We know birds are living dinosaurs, and their closest living relatives among non-birds are crocodiles.
Okay, so bracket dinosaurs between birds and crocodiles. What do birds and crocs share?
Well, both have four-chambered hearts. They both sing or vocalize to communicate. And significantly, they both build nests and show parental care, including brooding.
Like brooding, like sitting on eggs to keep them warm.
Essentially, yes, or otherwise caring for them with body warmth. Birds do it. Crocodiles cover eggs with vegetation or their necks. So phylogenetic bracketing predicted that non-avian dinosaurs likely did too. Built nests, cared for eggs, maybe even brooded.
And then they found the fossils.
Exactly. Fossils like Oviraptor found literally sitting over a clutch of eggs in a posture just like a brooding bird. It was stunning confirmation of the prediction made purely from the phylogenetic relationships.
It really brings the ancient world to life. Okay, so we can map relationships, predict features, but what about when things happened? Can we put dates on these evolutionary splits, especially for things that don't leave fossils easily?
That's where another ingenious tool comes in, molecular clocks.
A clock based on molecules. How does that work?
It's a method for estimating the absolute time of evolutionary events. The core idea is that some genes or parts of the genome seem to accumulate mutations at a reasonably constant average rate over long periods.
So the more differences you see in a particular gene between two species.
The more time has likely passed since they diverged from their common ancestor. The number of nucleotide substitutions acts like ticks of a clock.
That's brilliant. But how do you, set the clock? How do you know the rate?
You need to calibrate it. Scientists plot the number of genetic differences between pairs of species against the divergence dates for those species that are known from the fossil record.
Ah, so you use known dates from fossils to figure out the mutation rate per million years, for instance?
Exactly. You establish that baseline rate from reliable fossil data points. Then you can apply that rate to lineages where fossils are scarce or absent to estimate their divergence times.
But do all genes tick at the same rate?
No, definitely not. And that's actually informative. It depends partly on how important the gene is. Genes essential for survival tend to evolve very slowly. Why? Because most mutations in them would likely be harmful and get eliminated by natural selection. They're highly conserved.
Whereas less critical genes.
Less critical genes or non-coding regions might accumulate more selectively neutral mutations, changes that don't really affect the organism's fitness. These can accumulate more steadily, closer to a constant rate.
Are these clocks perfectly reliable, though?
Not perfectly. Natural selection can sometimes speed up or slow down the clock for certain genes. And if you try to extrapolate way, way back beyond reliable fossil calibration points, say hundreds of millions of years, the uncertainty increases.
So how do scientists deal with that?
They often use multiple genes, hoping to average out any irregularities, and they try to calibrate clocks using rates derived from a variety of different taxes to get a more robust estimate.
Can you give an example of where this has been really impactful?
A major one is dating the origin of HIV, the virus that causes AIDS.
Using a molecular clock on a virus.
Yes. Researchers compared gene sequences from HIV samples collected at different times, including an incredibly valuable sample from 1959 stored in the Congo.
And they saw a pattern.
They saw that the virus sequences had accumulated changes in a relatively clock-like manner over those decades. By calculating the rate of change and extrapolating backward.
They could estimate when the pandemic strain HIV-1M first jumped to human.
Exactly. The estimates pointed to an origin somewhere around the 1930s, possibly even a bit earlier, like the 1910s, according to some models, long before the epidemic was recognized in the 1980s.
Wow. That's a powerful insight with huge public health implications, all derived from tracking tiny genetic changes over time.
It really highlights the reach of these evolutionary tools.
So it's clear our understanding keeps evolving. Even how we classify life at the highest level has changed dramatically, hasn't it?
Oh, definitely. For a long time, it was just two kingdoms, plants and animals. Simple enough.
Then it expanded.
Oh, yeah.
I remember learning the 5 kingdoms.
Right. That came about in the late 1960s. Monera for the prokaryotes, then Protista, Plantae, fungi, and Animalia for the eukaryotes. That recognized the fundamental prokaryote-eukaryote split.
But that's not the standard anymore, is it?
No, because genetic data, especially rRNA sequencing, revealed something profound. The prokaryotes in Kingdom Monera were actually incredibly diverse. Some prokaryotes differ as much from each other as they do from us eukaryotes.
So Monera was too broad a category.
Way too broad. It masked deep evolutionary divisions. This led to the current 3-domain system: bacteria, archaea, and eukarya.
Bacteria and archaea being the 2 distinct prokaryotic domains.
Correct. This system emphasizes that 2 of the 3 great domains of life are entirely single-celled prokaryotes. And it also means Monera is obsolete, and Protista isn't really valid either in a strict cladistic sense. It's a paraphyletic grab bag.
This really changes the picture of the tree of life. But there's another complication, isn't there? Something that messes with the simple idea of branches splitting.
Ah, yes. The fascinating phenomenon of horizontal gene transfer, or HGT.
Horizontal, meaning not parent to offspring.
Exactly. Genes moving sideways between organisms, even across different species or domains. This can happen through viruses carrying DNA, bacteria exchanging plasmids, or even fusions of organisms like the ancient endosymbiosis that gave us mitochondria and chloroplasts.
Wait, so genes can just jump between branches on the tree of life?
It seems they can, and maybe quite often, especially early in life's history and particularly among prokaryotes. Studies suggest huge portions of prokaryotic genomes show signs of past HGT.
How does that affect building phylogenetic trees? They assume vertical inheritance, right?
It complicates things significantly. If you build a tree based on one gene that was transferred horizontally, you might get a different pattern of relationships than if you use another gene that was inherited vertically.
So the tree might look different depending on which genes you look at.
Precisely. For instance, some genes involved in metabolism in yeast look more similar to bacterial genes, while their core RNA genes firmly place them with eukarya, closer to archaea. HGT explains these conflicting signals.
Is there a good example of HGT providing a real advantage?
There's a great one. The extremophile alga Galgeria sulphuraria. It lives in incredibly hot, acidic, toxic environments.
I managed that.
Turns out, about 5% of its genes were acquired horizontally from heat-loving bacteria and archaea living in the same environment. Those borrowed genes likely gave it the toolkit to survive those extreme conditions.
That's incredible. Evolution by borrowing.
It really forces us to rethink the metaphor. Maybe the early tree of life wasn't so much a cleanly branching tree as, well, a tangled web or network, especially at its base.
Wow. So it seems the tree of life isn't some static diagram we finished drawing. It's dynamic, it's complex, and we're constantly refining our view as we get more data. It makes you wonder, what other tangled webs or fundamental surprises are still out there waiting to be discovered in life's history?
That's the exciting part of biology. We hope this deep dive into phylogeny has shed some light on how scientists are untangling that history.
From the entire Last Minute Lecture team, thanks so much for joining us.